Welcome everybody we're relaunching our Insert Create seminar series, Protein Innovates, and so we'll be going over the next, this term, rest of this term, and into next term. So we'll meet, we'll send a schedule out really soon. This year it's obviously sponsored by Insert Create, but also the Canadian Agricultural Partnerships with a number of the pulse grower organizations across Western Canada. Thank you to those sponsors. I'd like to introduce Dr. Wu. He's a full-time professor and director of the Division of Food Science and Bioresource Technology at the Department of Agriculture, Food and Nutritional Sciences at the University of Alberta in Edmonton. He's globally recognized for his research program in food-derived peptides and protein sustainability. So we've worked together on a number of grant applications, and we'll continue to do so. So he's published over 250 peer-reviewed publications in international journals, which was a huge feat on its own. 15 invited book chapters, 3 edited books, over 250 presentations, and 10 patent or patent applications. Dr. Wu has served on a number of editorial boards, associate editor, in various capacities, and is actively involved in a number of professional associations, including AOCS, IFT, International Society of Nutraceutical and Functional Foods, and so on. So we're really lucky to have Dr. Wu here for our first webinar to kick off the season. And I'll pass it over to you, and we're really excited.
Thank you so much, Michael, for your kind of introduction. Can you hear me well?
We can hear you.
Okay, thank you. So my talk is talk about peptides. So I really happy to have a discussion to see how we can use peptide as a strategy to add value to food protein and to reach the full potential for human health. So in today's presentation, I'm going to give you a brief introduction about peptides, and I'm going to present a 3 case study that about the peptides from cereal protein, from canola protein, and from pea proteins, and followed by some discussion and Q&A. Peptide present widely in nature. If we think of the first very important line of defense, defensing, for example, that present in wide range of animal, plant, even microorganism. In the snake venom that present as a toxic to human, that are also made of peptides. And even for the bacteria, I think we might have heard about the bacterium, which is one example is niacin is used as a food preservative. Is also bioactive peptides. And even in human, that range from control of food intake, appetite, and weight gain that is also controlled by a range of bioactive peptides. You may have heard about the significance of our stomach. If people can control your stomach, that's obviously is very important to be able to take care of the person. So the stomach, they can release a number of peptides that can affect your appetite and hunger and so on. And even in a cellular, for example, mitochondria, that contains a number of peptides as well. So one example that we can talk about is the 16 amino acid peptides we call MENC. So this tiny fragment of these peptides, they play an important role for messenger and also control homeostasis of glucose and weight gains. So as we can see, the key molecule in the body. Another example that play an important role in our body for controlling blood pressure also those small peptides. For example, angiotensin 2 is a vasoconstrictive peptide, can raise your blood pressure. And this peptide is formed by this enzyme we call angiotensin converting enzyme that use angiotensin 1 as this substrate. So increasing the level of angiotensin 2 will raise your blood pressure. That's why ACE is a widely used target for controlling your angiotensin 2 level, as well as to controlling your blood pressure. Peptides also present widely in a range of food commodities, such as in the fermented milk products, such as sour milk and cheese, and also in fermented meat products, such as ham. I presented some example in Huanju, which is a wine made from rice that contains about around 30% of peptides. Some even reported in the liquid also contains some peptides that may play some important role for the taste and some human health as well. So if we try to give a definition about what is peptides, we can simply say peptide is a fragment of a protein. So we present here one example as a fragment, as we highlighted in green, in the intact protein of lactoferrin. So these peptides can be released by using an enzyme that are present in our stomach, which is pepsin, that will generate as a mixture of these peptides. So if you look at this minimally inhibitory concentration of this wide range of hydroxylate comparison to this intact protein, so it's almost 10 times more potent in comparison to this intact protein, which indicates the significance of the peptides. So these individual peptides we call lactoferrin is a very well studied antimicrobial peptides that derive from food protein. Well, to prepare these peptides, we can obviously by using this any protein plus enzyme, and we can generate it by a mixture of peptides with some amino acids as well. So we can use the enzyme, we can use fermentation as we showed previously. And of course, some peptides that also present naturally in the food, range from animal to plants as well. Of course, including those fermented food as well. Well, as I mentioned, the pepsin that can release some lactoferrin from lactoferrin. So indeed, in our human body, we have a pepsin and we have a pyruvate and some peptides, and yes, indeed, by action of these protease, they can release some peptides in our body as well. We might have heard about it if we have this protein that as part of our diet, so they will break down by the enzyme and absorbed by the human for a variety of energy and as a substrate for synthesis of peptides or the protein. But during this digestion, as we can see, around 80% of this protein will break down in the form of a di or tripeptides. During this absorption, obviously because the action of the peptides, so majority of these di or tripeptides are first broken down into free amino acids, and then they are absorbed into our blood circulation in the form of amino acids. But still, we have around 10% of this protein that are absorbed in the form of small peptides, mostly in di or tripeptides. Indeed, if we look at literature, numerous bioactive peptides has been identified from a wide range of food, from milk, egg, animal protein, from cereal, pea protein, canola, and fish protein, and you name it. And these peptides are reported to be able to exert a wide range of bioactivity, range from antihypertensive, antioxidant, immunomodulation activity, and cancer, antimicrobial, anti-inflammatory, and others, of course, has some activity on metabolic syndrome, including glucose metabolism as well. So this slide is showing some example that were these peptides that are commercialized. So as we can see, most of these peptides are derived from milk. As we can see, those VPPI, PP are very well known. Others, whey protein hydrolyzate is mainly targeting these glycomacropeptides. Some others, as we can see, CPP are mainly for your mineral absorption. Others, of course, we see some from soy protein as well. Another is from fish protein. We recently published a book with my colleague Fido Pojo. So if anyone is interested in bioactive peptides, so you can look at this book chapter, this book, and which contains numerous research methodology and application of bioactive peptides. One very well-known example about bioactive peptides are those bioactive peptides that were identified from milk protein. So we call this lactose tripeptides, such as IPP and VPP. Well, these two peptides, as we can see, they present both in beta casein and cap casein, and they are released during fermentation. So these peptides are identified mainly as the inhibitor of this enzyme we call ACE. So as we mentioned earlier, inhibition of this enzyme is expected to reduce the angiotensin II level, which may have antihypertensive activity if they are bioavailable. And indeed, feeding the soy milk, as we can see, to this animal at 5 ml per kilogram body weight, reduce blood pressure significantly. Adding feeding these peptides, individual peptides, in equivalent the content in the soy milk also reduce the blood pressure in a comparable activity as that of soy milk, indicate the VPP and IPP are the responsible peptides in the soy milk responsible for blood pressure lowering activity. Well, blood pressure, as we mentioned, is mainly controlled by renin-angiotensin system. While our knowledge indicate activation of this renin-angiotensin system also lead to obesity and metabolic syndrome, which indicated the possibility if we reduce the activity of renin-angiotensin system, we may contribute to reduce the obesity and improvement of glucose intolerance. So give this consideration. So we did some research and trying to understand if these lactose tripeptides may provide a beneficial effect on metabolic syndrome. So in this experiment, we are using adipose tissue as a target. So we know adipose tissue is not an innate tissue, where is a very active metabolic organ. On the other hand, in addition to store the dietary fat, they were able to secrete a variety of adipokines, which play an important role for your energy metabolism. So for the healthy and functional adipocytes that are essential for healthy energy metabolism, well, when this normal function of adipose tissue is disturbed, for example, in the obese individual, So they often has enlarged adipose size, which is enlarged the cells. This increase the size will reduce the capacity for storage of fat. So this fat will likely, move to other place. So we call this ectopic fat disposition. So which will lead to consequences such as insulin resistance. So we talk about this change of this adipose tissue. So if we look at this healthy fat cells, it's a lot smaller than those obese fat cells. In addition, there's also infiltration of the immune cells. So as we highlight by this pink color, so the infiltration of these immune cells, mostly macrophage, will contribute to inflammation response. So we're not going to talk about this detail, but once the fat cell increase in size, that will release the fatty acids. So this release the fatty acids will eventually also contribute to that will be recognized by immune cells and will release these cytokines, will contribute to the inflammation. So that's why in obesity that also manifested by a chronic inflammation. So given this background, so once there's a dysregulated fatty acid metabolism, so that will lead to ectopic accumulation of fat in insulin sensitivity tissue, which lead to insulin resistance. So insulin is a hormone that responsible for glucose and the fat metabolism. So when this insulin action, for example, of the pre-adipocyte will lead to adipocyte differentiation that lead to smaller adipocyte cells, which increase the ability to store the fat that will reverse or improve insulin sensitivity. Well, we're trying to test if these peptides, they have a similar ability as the insulin to moderate the function of the adipocyte. So in this experiment, we're going to measure the adipocyte differentiation. So we look at these PPAR gamma and adiponectin release. And we also look in terms of the ability of fat storage by these fat cells as well. So when we look at the comparison with the peptide treatment as well as the insulin treatment, what we present here is an increasement in the ability of fat in the adipocyte, which indicate by treating the peptides, they show a comparable ability to enhance the ability of fat storage in adipocyte. So we also look at the ability to differentiate the fat cells. So as we can see that adipeptides show comparable ability to increase the release of adiponectin as well as the PPAR gamma, which are important biomarker for differentiation of pre-adipocytes. And we also look another marker, as we mentioned, as inflammation. We are looking for phosphorylation of P65. So as we can see, once we add the TNF alpha, which is a pro-inflammatory to trick the inflammation of the cells. Well, in comparison, as we add these two peptides, in particular added this VPP, we see a significant reduction in terms of this biomarker, so showing the ability of this peptide to reduce the inflammation. So from this research, we found that the milk-derived antihypertensive peptides, VPP and IPP, could have insulin-like activity at both sides, and also can suppress the cytokine-mediated inflammatory response. So in another study that are performed by colleague from Japan, so they look at the VPP in the animal if they can reduce the obesity associated insulin resistance and adipose inflammation. So the experiment is using these mice after 4 months of treatment, by two groups. One is control, one is VPP at a 1.1% based body this weight of diet, and then they look at the effect on glucose as well as insulin level. So this fasting glucose, fasting insulin level did not show any significant difference. Well, if we look at the C, if we look at the C group, which is by this insulin tolerance test, is a measurement of your insulin sensitivity. So in comparison to this control group, feeding VPP can significantly improve the insulin sensitivity. They also measured the cytokines expression in the adipose tissue. So a number of these cytokine markers, such as TNF-alpha, interleukin-1 beta, and the CCL5, showing significant improvement, indicate anti-inflammatory activity in the animal as well. They further look in terms of these adipose cells that in comparison to this control, adding this VPP, we saw reduced infiltration of the immune cells. So which is supportive in terms of reduced inflammation as we reported in the previous slides. So in a brief summary, the VPP, which is derived from milk protein, reduce infiltration of the immune cells, reduce the inflammation, of course, improve the insulin sensitivity in obese mice. Well, when we look at this serious storage protein, what is really interesting, if we look at the wheat, barley, oat, rye, maize, they also contain the same sequence of VPP and IPP in their primary sequence, which indicated the potential of using cereal protein to release those peptides that are, we just discussed from milk protein. By using fermentation, for example, our colleague, Dr. Marco Ganso, were able to determine VPP and IPP from the sourdough. So that's indicated by fermentation that we can release those peptides from those cereal protein. We did a structure function study, and we further identified some better sequence from cereal protein, such as IQP, VEP, IPQ, and VPE. And this slide is showing the identification of this sequence that from RAS protein. So we show the IQP relatively small, and VEP, and so on and so on. And this slide is showing that the content of these peptides that we can determine from this protein hydrolysis that are prepared from either rice or wheat, that's showing different levels. So we actually further did an animal study, similarly using a high-fat diet-induced obesity model, and we fed with these peptides to this animal, and we measure a number of the biomarker following this administration. So if we look at this oral glucose tolerance test, so if we compare to this high-fat diet, which is on the top, there's a trend of reduction, but not a very strong reduction. But there's a trend. But if we look at the area on the curve, obviously the VP had showed a significant reduction in the area. So we also look at this insulin tolerance test. What we did not see significant difference in this insulin tolerance test. When we look at the fasting glucose, on the other hand, we saw a clear trend of a reduction, as well as fasting insulin as well. And we look at this HOMA-IR, which is index measuring the insulin sensitivity. So we saw a significant reduction in comparison this high-fat diet, which support feeding this peptide can improve insulin sensitivity in this high-fat diet-induced mouse model. So similarly, we look at these fat cells. So this high-fat diet feeding, as obviously as we can see, enlarge the fat size as well as the infiltration of the immune cells. In comparison, when we fed these peptides, so there's a reduce the fat cells and also reduce the we do not see obvious infiltration of the immune cells. So similar to previous results. We also look at some peptides, for example, ACE inhibitory peptides from canola protein. So we show some sequence, valine, serine, valine, and phenylene and leucine that were identified from napping and crucifrine. Our colleague from Japan also identified some peptides from canola as well. So one of the example is, for example, is arginine, isoleucine, and tyrosine that are identified from this rapeseed or canola protein that are showing able to reduce blood pressure as well. So we continue this research trying to understand the mechanism action of these peptides. So as we mentioned earlier, that ACE, this enzyme is a key target for the blood pressure lowering activity. What is interesting, in the research is numerous peptides that are showing ACE inhibitory activity, not necessarily that showing ACE inhibition in vivo. So this led to us trying to understand if some other mechanism, more specifically an ACE2, which is an analog of ACE, but play an important role by using angiotensin 2 as its substrate to produce angiotensin 1-7. Well, activation of ACE is detrimental to our health. On the other hand, activation of ACE2 is believed to be benefits for the cardio health. So in this research, we're trying to see if feeding this peptide to this animal that may lead to activation of ACE2, which may explain a new mechanism of action of these peptides. Well, indeed, when we fed this peptide to the animal, we confirmed this peptide could reduce the blood pressure. What we saw, again, we did not see change of ACE activity, but we did see in terms of ACE2 activity was significantly increased, as well as the protein level was also increased, as well as the gene level was also increased. In alignment with this observation, we saw angiotensin 1-7 was increased, which is a product of action of ACE2. So this study indicates this RIY is a ACE2 activated in vivo that that is responsible for its antihypertensive activity. ACE inhibitory peptides were also identified from pea protein and soy protein. So in our early study by based the structure function study of ACE2 active ACE inhibitory peptides, so we identify a number of the sequence, for example, FW from garden P, WW from soybean, and we also look at here LRF from bovine milk, LRW from pea protein. Well, I wanted to highlight that this LRW that's showing a lot of low log IC50 value, which is an indication of very strong activity of these peptides. So IC50 is an index we trying to represent the ability of a compound to inhibit the activity of enzyme. So obviously the low activity indicates the strong activity of these compounds. So as we can see, this LRW is a lot smaller than this VPP and IPP, which are two peptides that we mentioned a few times from milk. So this peptide is supposed to be extremely strong in the activity. Previously, my lab identified peptides IRW from egg protein of transferrin and showing very strong antihypertensive activity in the animal. So if you look at this ARW, which was identified from pea protein, so they are actually analogous that only differ by the first amino acid. So we look at their IC50 value as I just mentioned. So as you can see, the LRW is a lot strong in terms of ACE inhibition than IRW. So we look into this blood pressure lowering activity. To our surprise, we do not see any blood pressure change after feeding these peptides at the 50 milligram per kilogram body weight to the animal. So the SBP, MAP, DBP, there's no change at all. So we also look at the heart rate. We did not see change in the heart rate, so which indicated the heart rate will not affect the blood pressure really in the experiment. So next, we also look at the components of the renin-angiotensin system. We look at ACE, there's no difference for ACE2, no difference for the AT1 receptor, AT2 receptor, which is a receptor for angiotensin 2. We also look at the receptor for muscle receptor. There's no difference as well. So obviously, this peptide will not affect the renin-angiotensin system. And we also look at the inflammatory biomarker. We look at a number of the biomarker in the VCAM1, ICAM1, which are adhesion molecule that important to bind to the immune cells. So we do not see difference. Similarly, we do not see difference in the inos as well as the COX-2 that are responsible for the formation of inflammatory molecules. So next, we try to understand if this lack of activity of these peptides is because of lack of bioavailability. So we first perform an in vitro experiment to look at the stability against the gastrointestinal enzyme pepsin and pacritin. So these two peptides, as we show here, are stable against the pepsin, but they show very different stability against the pacritin. As we can see, that LRW that left only around 1%, but IRW that's showing around 52%. So showing majority of this LRW is likely broken down by the action of the peptide, but mostly likely by this trypsin or chymotrypsin, the percent in peptide. So by using HPLC, we further confirm these results. And we next look at about the stability of this peptide during uptake by epithelial cells. So we are using CAC2 cell as a model to measure the stability of these peptides. In comparison, as we can see, this IRW show better stability than this ARW. So in the experiment, we added the sample in the apical site, and with the peptides pass through this monolith in this basolateral side, and we take the sample that from both sides for analysis. So as we can see in this apical side, so we can this basal side we see a significant reduction in this LRW in comparison to IRW, which indicate the ability of this peptide to be uptaken by epithelial cells is significantly less than this IRW. So this again support this peptide is possibly not able to pass through the epithelial cells to exert its biological function. So this indicator, I think, that the peptides in the P protein, so we were not able to confirm the activity of ARW, even though it is showing promising in vitro activity. So further research, we show that other candidates, IKP, YW, and FW, for their ability to reduce the blood pressure. In terms of the bioavailability that we talk about, this LRW is low in comparison with the experiment. For the IRW, actually we see time-dependent increase in the peptide level in the animal in the 20 minutes experiment. We are going to continue some study in the human. Well, the next key question we wanted to discuss for this presentation is really what makes food protein good candidates for preparing bioactive peptides. So in the literature, we see a lot more research, those peptides from milk, but not necessarily always the case. So the protein demand we all understand is on the rise. So the protein obviously play an important role as an energy, as an essential amino acids. While protein are also acclaimed for their ability to sustain your energy, to improve your mental health, improve your joint and bone health. Well, we all understand that the animal protein is generally speaking is high in terms of quality than plant protein. Well, in addition, they contain a much high protein content, except soy protein, than the rest of other plant protein. Plant protein also considered as incomplete because they are deficient always in one or more of those essential amino acids. They contain inhibitors of protease. They also contain some secondary metabolites which might interfering protein metabolism. So that's make plant proteins are inferior in terms of quality. In assessing the quality of protein, I think people tend to look in terms of their level of essential amino acids, their bioavailability, of course, the digestibility as well. And then we tended to classify the quality of protein largely based the essential of amino acids profile. From this presentation, we show a number of peptide sequence that we saw in milk, we saw in egg, and we also identified there are the same sequence or compatible sequence from cereal protein, from canola, from pea protein. So when we try to looking for this protein to propel bioactive peptides, it seems not always the case that high quality protein are better candidates for preparing bioactive peptides, which I think is really a good thing, as that gives us a lot of opportunity to make good use of plant protein. So some potential that we are working on for plant protein is rice protein, mostly from those byproducts, such as rice bran or broken rice that are produced during processing, or gluten that is considered byproducts from wheat starch processing, or the brewing industry, for example, generate millions tons of what we call brewer's spent grain. So there also contains substantial amount of protein that can be used for the formation of bioactive peptides. The canola industry that is very important for supply oil, to the Canadian. That they're also a major source of protein, even though there are some application in the food, but as we mentioned, they also provide a lot of opportunity for propel peptides from canola. So the similar story about the pea protein, obviously the demand for the pea protein is growing substantially, and this pea protein processing also produce a great amount of byproducts that can be used for making peptides as well. And I really wanted to acknowledge the support from a number of the founders that as well as the industry that support my research. And I also wanted to highlight, acknowledge my students and the trainees. They are supported in this research, and also wanted to thank you for your attention, and happy to take any question that you may have. Thank you.
Great, thanks very much, Jianping. Can you hear me okay?
Yes, I can hear you very well. Thanks, Jim.
Yeah, Dr. Nickerson had to step away to deal with an issue, so I'll manage the question period for the rest of the session, if that's okay. Excellent presentation. Thank you for your questions. I'm going to open this question up now, or the floor up now to questions. I wonder if you could reduce the screen so I could see if people are raising their hands. There we go. And let's see if we've got, so the floor is now open for questions, and I'd like to encourage our faculty members and our students to ask questions. You just use the raise your hand button. I'll get you started. What do you see as some of the biggest hurdles that you've had to face with moving forward with bioactive peptide research? Maybe just as a bit of a learning experience for some of the students and the trainees in the group.
Very good question. So by the way, I'm trying to finish this presentation in about 45 minutes, so we can have more time for discussion. So you are asking a very challenging question. Like, to me, I think one of the hurdle to move these peptides for commercial application obviously is lack of evidence to demonstrate the efficacy and safety of these peptides. A lot of people, talk about the peptides, obviously come up to the question, they are not stable, they are going to break down. Hey, wait a minute, I think in the body we have so many peptides that function as a hormone that regulate a wide range of biological function in our body. So we have to generate a lot more evidence to support the efficacy of as well as safety of peptides for functional food application.
So that sort of leads into another question about sort of the regulatory process involved in moving forward functional foods and nutraceuticals or natural health products into the marketplace. So you definitely need safety and efficacy data. Are there any other hurdles that, in terms of maybe intellectual property or the classic one of identifying funding sources, right? That to assist with the research. Right?
Well, I agree. I think peptides is now immune to this regulatory hurdle in the IP hurdle and the industry engagement. So I think any hurdle that we might have heard about from other functional food, phytochemicals, dietary fiber, beta glucan, we may see similar challenge as well. We actually working with the Health Canada right now in terms of this clinical trial, and I think we are in discussion how we're going to regulate these peptides. Shall we list as a food or as natural health products? Obviously, I think their response is kind of vague, but obviously there's no clear regulation of peptides in Health Canada, so there's a need more research to define how to regulate the peptides by Health Canada.
Great. Well, I've started the ball rolling. I'm looking for our students and our faculty to get engaged with asking questions, or I'm going to start calling and then watch everybody click off. We've got an excellent presentation. Do we want to? I'm going to open the floor up to questions again from folks. I see Ningen is turned your camera on. So your colleague from the U of A would like to ask a question. Go ahead, Ningen.
Yeah, I think we also have some work about a peptide from plant proteins, but a question normally challenging ours is that you can generate this sequence from many proteins. So then, right, so sometimes you can do research about one source of protein to generate this kind of peptide, and then next day you can generate peptide from another source of proteins. So, I mean, you also have the last slide to say what kind of protein would be good to generate a peptide. I would like to see if you have more in-depth consideration of that question.
well, I think that's obviously, I think, very interesting conversation, when I started my research on peptides, I think back to many years, I don't know, like over 20 years ago, the people were asking why you choose this protein but not other protein, right? Well, similarly, why you choosing this enzyme, not the other enzyme? Similar question. So indeed, I think if we look at this availability, there are so many proteins, ranging from animal, plant, fish. We have some, right now, this single cell proteins, second proteins. Also, the option is almost endless. I think, if we look at the literature, when challenges, we see a lot more research on peptides from dairy protein. Which is something I was trying very hard to say, trying to convince. The reason there's a lot more research on dairy protein is because there's a lot more research going on that are looking for the peptides from milk protein. But this does not indicate the milk protein are the best protein for producing bioactive peptides. I think I did a pretty good job for the past 10, 15 years that convinced people egg protein are greater protein for producing peptides. So, in the same line, I'm trying to convince people that the plant protein often considered as inferior protein in comparison to animal protein. They are actually pretty good as well to make bioactive peptides. So I hope by presenting the presentation today that we're showing some argument, not necessarily conclusion, that we can use the cereal protein, we can use canola protein, we can use horse protein as the material to produce the peptides with comparable, even better activity than those peptides from milk protein. So I see that's obviously can open up our window for the industry can harvest the opportunity for value addition. And I think that it might also have relevance for human as well, because we almost rely the essential amino acids as the health nutrition benefits of protein. I would argue that's not is not the whole story. Those non-essential amino acids, they also play an important role in the human health as well. So in certain degree, I even disagree that we should classify essential versus non-essential amino acids. This is the same argument I often have with primary versus secondary plant metabolites as well, because they are essential for the survival for plant as well. I'm not sure that answered your question or not.
Yeah, thank you, Jianping. This is very good insight. I also like your argument that the high quality protein with high essential amino acid may not necessarily be the high quality protein for peptide. So thank you, Jianping, for share of your thought.
I think it's very interesting because Jim has obviously expert in protein quality. so that's yeah.
Well, quality you can define many ways, and I think looking at it from just the classic definition, you brought it that forward, I think, but you really opened up the eyes to the alternative concepts of quality, and I guess that's at the heart of one of the questions I have is: do you see that you're generating bioactive peptides by starting with the source of protein and digesting it, and then isolating those peptide fractions? Is there a sufficient generation of bioactive peptides just if you're eating whole foods? Is it enough, or is the dose too small?
Really good question. So I think I should be cautious to answer your question without much detail and the context. I would say, I think, as I showing the example, even we eat food protein, so we still have around 10% that may reach our circulation as small peptides. If we use say like a food as an example, so in our body we have this digestive enzyme, so they are very specific, they are very efficient, saying that they can release the peptides. We can almost predict what it looks like. But by using fermentation or enzymatic treatment in vitro, that impart our ability to generate the sequence that may not generated by our body. So they may have different sequence, and so they may have different activities. So, well, of course, I think even if we eat a peptide protein, we may generate some sequence.
Okay, yeah, I like that answer in terms of you're providing a measure of control over the system through precision fermentation or enzymatic hydrolysis to generate the actual peptides with the bioactivity. We have opportunity for a few more minutes of questions. I'd like to open up this floor again for students or faculty to ask questions of Dr. Wu.
No,
we've got a quiet group. Yeah, quiet group today.
Yeah.
I'll throw it to one of our faculty members. I see Dr. Luco's online. Anything that you'd like to ask your old colleague about? You gotta see if he's paying attention. I'll see if he's paying attention.
Not really. I mean, I think he's covered most of the bases. You know, most of the time, I think the question one also gets a lot is, you know, the rationale for the choice, right, of proteins, you know, and my answer most of the time is, you know, it depends on where you're working, and it depends on, you know, what will make sense economically to the area, because if I'm in the prairies, it makes sense for me to work on cereals and legumes and oil seeds than to work on fish or, you know, stuff like that. So, but in general, I think he covered all the areas. So, great job.
Thank you, Rotimi.
Yeah, well, thanks, Rotimi. Sorry to put you on the spot. That was good to hear your perspective there. I think one of the terms that we hear a lot about is so the circular economy and how we can upcycle foods that are going into the waste stream, and certainly the generation of value-added products, and bioactive peptides would certainly fit within that category. You've done some work on some co-products from the processing industry. What's been some of your experiences there in terms of opportunities moving forward for adding value to some of the processing sectors, be it from the animal side or even on the plant side?
So that's a question for me?
Sure, yeah.
Right.
Yeah, I think Rotimi's gone now, so you can't put it back to him.
My apologies. I think I thought that the presentation, you know, should be 1 hour late. I didn't realize we have 1 hour difference. I haven't refound, you know, the slides. I think so that I went a little bit faster. Great question, Jim. So my lab, I think we are working on a wider range of upcycle bioproducts from the industry. So from animals bioproduct side, so not necessarily on eggs. We also working on spent hens, and we also working on the feathers as well. From the plant side, we are working on canola pods, as well as spent hen, spent grain as well. I saw Nadika's question.
Is there a question there from yeah, Nandika, go ahead. Thank you.
I don't often get chance to ask questions from Dr. Wu, right? So I take this as an opportunity to ask a question from him. So, okay, no, like in terms of using the bioactive peptide, like do you see more opportunities as, let's say, supplement, or like going through that nutraceuticals route, or like adding it as, let's say, food additive or food ingredient in food formulation? Like which part would have a better opportunity in terms of short-term actual usage in like for given bioactivity that you discussed?
But you know, very important question. So you know, obviously to the end, we have to find a way how to deliver the benefits of peptides to the consumers, that in a way that a consumer that feel comfortable. So I think the best way, obviously, trying to look the food that the consumer enjoy already. So there's no way to introduce some new, you know, food products to the consumer. So my personal preference, I would love to use those peptides as a food in the format such as, you know, those high protein beverage, which is highly liked by numerous consumer. You know, adding those peptides into food products obviously is also attractive, but you know, the interaction effect of processing and possibly the sensory attributes may complicate the consumer acceptance. So I think using these peptides as natural health products, which seems, I think, has Canada from my discussion with them as possibly another way that for use as well. So similarly, you know, if we're using collagen as an example, So collagen peptides in Canada is regulated as natural health products. So I see those peptides that, could be regulated as natural health products as well in Canada.
Okay, now the reason I ask is like if you add it into, let's say, food product, then the processing impact would come into play, right? Like whether the peptide would have the same bioactivity after processing. So if it is a product that does not go through like a severe processing, that's fine. But most of the time we do use some sort of process. So that's why I want to know like whether that would be the best part.
Yeah. I think we can just sell those powder and people can just make a shake, protein shake, right? As a high energy boost as well as to have some benefits will be also nice as well.
Okay, thank you.
Thanks, Nandika. Well, we've reached the top of the hour. I want to just take this opportunity to thank you, Dr. Wu, and for presenting an excellent presentation. You gave us an excellent overview of your research and positioning it within the context of bioactive peptides. So please join with me, everyone, to thank Dr. Wu for just an excellent presentation. You can show your appreciation through the reactions buttons at the bottom, and we'll all give a clap there.
Oh, thank you so much.
So thank you very much.
Yeah. Let me know if you have any question and email me or whatever you feel comfortable. Okay. Have a good day. Bye-bye.
Okay. Well, thank you very much and have a good afternoon, everyone. Enjoy the rest of your day.